Welcome friends. Today I am going to deliver a lecture on microbial nutrition. The main objectives of this lecture will be to understand the common nutrient requirements of microbes, requirements for carbon, hydrogen, oxygen, nitrogen, phosphorus and sulfur, nutritional types of microorganisms, growth factors, culture media and types of media, and finally I will talk about isolation of pure cultures. Dear students, talking about the common nutrient requirements of microorganisms, I would like to tell you that microorganisms, like all other living organisms, require different types of nutrients for their growth. Analysis of microbial cell composition shows that over 95% of cell dry weight is made up of a few major elements like carbon, oxygen, hydrogen, nitrogen, sulfur, phosphorus, potassium, calcium, magnesium and iron, called macronutrients or macroelements, because they are required by the microorganisms in relatively large amounts. The first 6 compounds, which are carbon, hydrogen, oxygen, nitrogen, sulfur and phosphorus, are common components of carbohydrates, lipids, proteins and nucleic acids, and the remaining 4 elements exist in the cell as cations and play a variety of roles. For example, potassium is required for activity by a number of enzymes, including some of those involved in protein synthesis. Calcium, among other functions, contributes to heat resistance of bacterial endospores. Magnesium serves as a cofactor for many enzymes, complexes with ATP and stabilizes ribosomes and cell membranes. Iron, both Fe2 positive and Fe3 positive, is a part of cytochromes and a cofactor for enzymes and electron carrying proteins. First and foremost, the requirements for carbon, hydrogen, oxygen, nitrogen, phosphorus and sulfur. The requirements for carbon, hydrogen and oxygen often are satisfied together. Carbon is needed for the skeleton or backbone of all organic compounds and molecules, serving as carbon sources, normally also contribute both oxygen and hydrogen atoms, thus becoming a source of all 3 elements. Carbon sources frequently serve as energy sources, although they do not have to. One important carbon source that does not supply hydrogen or energy is carbon dioxide. This is because carbon dioxide is oxidized and lacks hydrogen. Probably all microorganisms can fix carbon dioxide, that is, reduce it and incorporate it into organic molecules. However, by definition, only autotrophs can use carbon dioxide as their sole source of carbon. Many microorganisms are autotrophic, and most of these carry out photosynthesis and use light as their energy source. Some autotrophs oxidize inorganic molecules and derive energy from electron transfers. Organisms that use reduced organic molecules as carbon sources are heterotrophs. For example, the glycolytic pathway produces carbon skeletons for use in biosynthesis and also release energy as ATP and NADH. Nitrogen is needed for synthesis of amino acids, purines, pyrimidines, some carbohydrates and lipids, enzyme cofactors and other substances. Many microorganisms use the nitrogen in amino acids, and ammonia often is directly incorporated through the action of such enzymes as glutamate dehydrogenase or glutamine synthetase and glutamate synthase. Most phosphorus and many non-photosynthetic microorganisms reduce nitrate to ammonia and incorporate the ammonia in assimilatory nitrate reduction. A variety of bacteria, for example many cyanobacteria and the symbiotic bacterium Rhizobium, can reduce and assimilate atmospheric nitrogen using the nitrogenase system. Phosphorus is present in nucleic acids phospholipids, nucleotides like ATP, some proteins, several cofactors, and other cell components. Almost all microorganisms use inorganic phosphate as their phosphorus source and incorporate it directly. Low phosphate levels actually limit microbial growth in many aquatic environments. Sulfur is needed for the synthesis of substances like the amino acids, cysteine and methionine, some carbohydrates, biotin and thiamine. Most microorganisms use sulfate as a source of sulfur and reduce it by assimilatory sulfate reduction. A few also require a reduced form of sulfur such as cysteine. Now, the nutritional types of microorganisms. Microorganisms can be grouped into nutritional classes based on how they satisfy all the requirements. With respect to their preferred source of carbon, microorganisms can be classified either as heterotrophs or as autotrophs. There are only two sources of energy available to the organisms. Number one, light energy, and number two, the energy derived from oxidizing organic molecules or inorganic molecules. Phototrophs use light as their energy source. Chemotrophs obtain energy from the oxidation of chemical compounds, either organic or inorganic. Microorganisms also have two sources for electrons. Lithotrophs, that is rock eaters, use reduced inorganic substances as their electron source, whereas organotrophs extract electrons from the organic compounds. Despite the great metabolic diversity seen in microorganisms, most may be placed in one of the four nutritional classes based on their primary sources of carbon, energy and electrons. The large majority of microorganisms thus are either photolithotropic autotrophs or chemoorganotropic heterotrophs. Photolithotropic autotrophs, often called photoautotrophs or photolithoautotrophs, use light energy and have carbon dioxide as their carbon source. Chemoorganotropic heterotrophs, often called chemoheterotrophs, chemoorganoheterotrophs or even heterotrophs, use organic compounds as their source of energy, hydrogen, electrons and carbon. The other two nutritional classes have fewer microorganisms but often are very important ecologically. These photoorganotropic heterotrophs, photoorganoheterotrophs are common inhabitants of polluted lakes and streams. Some of these bacteria also grow as photoautotrophs with molecular hydrogen as an electron donor. The fourth group, the chemolithoautotropic autotrophs or chemolithoautotrophs, oxidizes reduced inorganic compounds such as iron, nitrogen or sulfur compounds to derive both their energy and electrons for biosynthesis. The microbes that combine chemolithoautotropic and heterotropic metabolic processes are called mixotropic. Now, I will talk about the growth factors. Organic compounds which are required by the microorganisms because they act as essential cell components or precursors of such components and are not synthesized by the organisms are called growth factors. There are three major classes of growth factors. Number one, amino acids. Number two, purines and pyrimidines. And number three, vitamins. Amino acids are needed for protein synthesis, purines and pyrimidines for nucleic acid synthesis, and vitamins are small organic molecules that usually make up all or part of the enzyme cofactors. The specific growth factor requirement of different microorganisms makes quantitative growth response assays possible for a variety of substances. For example, species from the bacterial genera Lactobacillus and Streptococcus can be used in microbiological assay of most vitamins and amino acids. The appropriate bacterium is grown in a series of culture vessels. each containing medium with an excess amount of all required components except the growth factor to be acid. A different amount of growth factor is added to each vessel. The standard curve is prepared by plotting the growth factor quantity or concentration against the total extent of bacterial growth. Ideally, the amount of growth resulting is directly proportional to the quantity of growth factor present. If the growth factor concentration doubles, the final extent of bacterial growth doubles. The quantity of the growth factor in a test sample is determined by comparing the extent of growth caused by the unknown sample with that resulting from the standards. Dear friends, now let us discuss the culture media. A culture medium is a solid or liquid preparation used to grow, transport and store microorganisms. To be effective, the medium must contain all the nutrients the microorganisms require for their growth. Specialized media are essential in the isolation and identification of microorganisms, the testing of antibiotic sensitivities, water and food analysis, industrial microbiology and other activities. Although all microorganisms need source of energy, carbon, nitrogen, phosphorus, sulfur and various minerals, the precise composition of a satisfactory medium will depend on the species one is trying to cultivate, because nutritional requirements vary so greatly. Knowledge of a microorganism's normal habitat often is useful in selecting an appropriate culture medium, because its nutrient requirements reflect its natural surroundings. Frequently, a medium is used to select a growth-specific microorganism or to help identify a particular species. In such cases, the function of the medium also determines its composition. Despite the fact that microbiologists have been growing microorganisms in laboratory cultures for over 125 years, most microorganisms in nature are yet to be cultured, leaving many challenges to the microbiologists today. Common constituents of culture media are water, sodium chloride and other electrolytes, water-soluble peptone, yeast extract, carbohydrates and a solidifying agent called as agar. Now, the types of media. Culture media can be classified on the basis of several parameters, like the chemical constituents from which they are made, their physical nature and their function. Types of culture media based on chemical composition. Based on chemical composition, there are two broad classes of culture media. Number 1, defined media, and number 2, the complex media. Defined media or synthetic medium is a medium in which all chemical components are exactly known. It can be in a liquid form or solidified by an agent such as agar. Defined media are often used to culture photolithotropic autotrophs such as cyanobacteria and photosynthetic protists. Defined media are used widely in research, as it's often desirable to know what the experimental microorganism is metabolizing. Some defined media, such as the one listed for Escherichia coli, are said to be simple because they contain only a single carbon source. Complex media. Media that contains some ingredients of unknown chemical composition are called as complex media. Complex media employ digesters of microbial, animal or plant products such as casein, that is milk protein, beef, that is beef extract, soybeans, that is tryptic soy broth, yeast cell extract, or any of a number of other highly nutritious yet impure substances. These digesters are commercially available in dehydrated form and can be easily prepared. Three commonly used complex media are: number 1, nutrient broth, number 2, tryptic soy broth, and number 3, McConkey agar. Functional types of media. Number 1, basic media. These are simple media such as nutrient agar, nutrient broth or peptone water that supports the growth of microorganisms that do not have special nutritional requirements. They are often used in the preparation of enriched media. Number 2, enriched media. An enriched media is a broth or solid medium containing a rich supply of special nutrients, for example, blood, serum or egg, that promotes the growth of fastidious organisms. Commonly used examples are blood agar and chocolate agar, both of which are rich in nutrients and free from inhibitory substances. Number 3, the selective media. Selective media are the media that favor the growth of particular microorganisms. Endo-agar, Eosin methylene blue agar and McConkey agar are 3 media widely used for the detection of Escherichia coli and related bacteria in water supplies and elsewhere. These media contain dyes that suppress gram-positive bacterial growth. Number 4, differential indicator media. Differential media are media that distinguish among different groups of microbes and even permit tentative identification of microorganisms based on their biological characteristics. Blood agar is both a differential medium and an enriched one. It distinguishes between hemolytic and non-hemolytic bacteria. Hemolytic bacteria, for example, streptococci and staphylococci isolated from throats produce clear zones around their colonies because of red blood cell destruction. McConkey agar is both a differential medium and a selective medium. Likewise, Kliger iron agar and triple sugar iron agar both differentiate between different types of Enterobacteriaceae on the basis of their ability to ferment carbohydrates. Number 5, selective and differential media. An example of this is McConkey agar, which is selective for enteric and other gram-negative bacteria, while inhibits gram-positive bacteria and has an indicator neutral red to differentiate between lactose and non-lactose fermenting microorganisms. Another example, that is mannitol salt agar, is selective and differential to grow Staphylococcus aureus. Types of culture media based on physical nature. Liquid culture media are sometimes solidified by the addition of a gelling agent. Solid media immobilize cells, allowing them to grow and form visible, isolated masses called colonies. Solid media are prepared in the same way as liquid media, except that before sterilization, agar, a gelling agent, is added to the medium, typically at a concentration of 1 to 2%. The agar melts during the sterilization process, and the molten medium is then poured into sterile glass or plastic plates and allowed to solidify before use. Finally, I will talk about isolation of pure cultures. Most specimens from animal tissues, plant tissues or environmental samples are mixed with a variety of bacteria or other microorganisms. A single gram of feces, for example, has over 10 raised to the power 10 bacteria, and that gram would have over 20 different bacterial species. Particularly in a medical setting, where a patient's body fluid or tissue, skin, blood, spinal fluid or urine is sent to the microbiology laboratory for analysis, the specimens most often are mixed in order to identify the bacteria and run antibiotic susceptibilities on the causative agent of the infection. The microbiologist is bound to work with a pure culture of the bacterium. The exercise begins with a mixed culture of bacteria and ends with, hopefully, pure culture of the microbe. A pure culture theoretically contains a single bacterial species. One needs a pure culture, a population of cells arising from a single cell, to characterize an individual species. Pure cultures are so important that the development of pure culture techniques by the German biologist Robert Koch transformed microbiology. Within about 20 years after the development of pure culture techniques most pathogens responsible for the major human bacterial disease have been isolated. There are a number of procedures available for the isolation of pure cultures from mixed populations. A pure culture may be isolated by the use of specialized media with specific chemicals or physical agents that allow the enrichment or selection of one organism over the another. Simpler methods for isolation of a pure culture include: number 1, spread plating on solid agar medium with a glass spreader, and number 2, string plating with a loop. The purpose of spread plating and streak plating is to isolate individual bacterial cells, colony forming units on a nutrient medium. With this, we conclude. Thank you for watching.